So today we're going to talk about adaptive immunity and organ transplant rejection. So each year around 40,000 Americans are saved by organ transplant, and in 1954 that's when the first successful human kidney transplant was performed, and that's a really big breakthrough in medicine, but transplant attempts before and after weren't so lucky. There were symptoms such as pain, fever, shortness of breath, organ failure, and even death. And to understand why this happened, we first need to understand adaptive immunity, but first the general immune system. So the job of the immune system is to protect the body from foreigners such as bacteria and viruses. There are three breakdowns: innate immunity, passive, and adaptive, which we are going to focus on today. So adaptive immunity, we basically develop it through life as we are exposed to pathogens either naturally or through vaccination. The characteristics of the adaptive immunity are a slower and more specific response, a diverse range of molecules that it can respond to, memory for faster repeated response, and the ability to distinguish between self and non-self. So the key players you need to know are first antigens, which are like bacteria, virus, toxins, or even a disease cell that the body recognizes as non-self. Antibodies, which are proteins produced in response to a specific antigen that circulate the body. B cells, which make antibodies, and T cells, which are activated by foreign antigens and kill target cells. So here are the players specifically for organ transplant rejection. So blood antigens are what they sound like. They're basically free-floating antigens in the blood. And the next thing is really important is the major histocompatibility complex, which is basically glycoproteins on each cell that act as a tag that say, "This is me." So the white blood cell will read this tag, and if it matches, it won't attack the cell. And you need these because it provides histocompatibility between the organ donor and the recipient. The problem is that there are 20 plus genes that code for MHC, and there are 50 plus combinations per gene, which leads to lots of variations, and it's very hard to match a donor and recipient. So if you remembered me talking about the first successful kidney transplant, that was actually what made the kidney transplant successful, because it was performed on identical twins who had identical antigens and their MHCs matched. So the organ was accepted. So now we just have a quick little demonstration showing what happens when an organ is being rejected. Let's say this cupcake is a recipient cell and these cupcakes are donor cells. You can see that the MHCs between the recipient and this donor match, so the kidney is accepted. The MHC doesn't match this donor though, so the kidney is rejected and the cells are killed off by T cells. Same applies to blood antigens. The antigens of this recipient and donor are the same color and they match, so the kidney is accepted. This isn't the case for this donor, so it is rejected. So now we'll go through the types of rejection. Just remember, rejection is when the organ is identified as foreign, so the body kills the cells and rejects it. So the first is hyperacute. This occurs minutes to hours after the organ is put in. Doctors actually will wait 10 minutes to see if the organ starts changing color, and if it does, they'll take it out, and this is due to preformed antibodies. The next is acute rejection. This happens weeks to months after the transplant. There are two categories. One is humoral, which is when free-floating antibodies cause the attack of the organ, and then the next is cell-mediated, where cytotoxic T cells go in and specifically target the organ cells to kill them. This can actually be controlled, they realized in the 1980s, by immunosuppressants that suppress the immune system, such as cyclosporine, but there are different drawbacks. So the patient, throughout their life, they have to be on a large number of drugs, almost 12 pills a day, and because it weakens their immune system, they're more subject to other diseases such as infection and cancer. And then the last is chronic infection, which happens months to years after transplant, and it's the same antibodies and cell-mediated response seen in the acute rejection, results in fibrosis, scar tissue, and then loss of the organ. You can probably tell by now that rejection is quite a big problem, and immunosuppressants aren't really good enough solution, so people are actually looking at 3D printing an organ, and before you're like, oh my god, what? I'll explain the science behind that. So basically they use bioprinting, which is 3D printing technology using living cells to produce tissues. So I found a case study explaining how they're testing this, and basically why it works is because if you're using a recipient's own cells to create the organ, the body will identify the organ as self, and there's virtually no chance for rejection, and so you can eliminate the troubles of immunosuppressants, and it's a very promising solution for the future, which I thought was really interesting and a good way to close this lesson.